I would like now to introduce our next rising star speaker, Dr. Gill. Sar obtained his medical degree from the University of Melbourne in Australia in 1999 and subsequently trained in internal medicine and hem-onc before moving to Stanford, actually to Rob Negrin's lab as a postdoc in 2008. In 2011, he moved to UPenn for BMT in cellular therapy fellowship, and since 2012 holds a faculty position. He's currently an associate professor of medicine and scientific co-director of the cell therapy and transplantation program at UPenn. In addition to all his academic activity, he has co-founded 2 biotech companies within the last decade. He has made significant contribution to the CAR field, focusing on CAR T-cell therapies for hematological malignancy, in vivo delivery of CAR encoding genes, and the use of macrophages as a cell therapy platform for CARs. Please extend a warm welcome to Dr. Gill, who will speak to us today about CAR-redirected macrophages for the treatment of solid tumors.
Thanks very much for the introduction, Dr. Gottschalk. Well, thank you. I'm very honored to be invited for this Rising Star seminar series. So I do have some disclosures, as Dr. Gottschalk pointed out. I have founded 2 companies. One of them is relevant to this presentation because I founded it in order to translate to the clinic as quickly as possible our findings on CAR macrophages. That company is called Charisma Therapeutics. The therapeutic landscape of CAR T cells, as published at the start of last year, is indicated here. This is a really nice paper that I think did a really good job of laying out the clinical trial landscape, not preclinical, but clinical trial landscape around the world in liquid and so-called solid malignancies. This heat map shows where the squares are white indicates no clinical trials, and then with the heat map up to a brown color, a large number of clinical trials being conducted in the indicated malignancy, as indicated on the left, the vertical axis of this graph. And while I'm a practicing hematologist and most of my clinical work is in liquid malignancies, I was kind of intrigued to start playing in that very white sparse field over on the right, which is the solid malignancy space, where you can see that there are relatively few cancers that are being currently being tested clinically with the CAR T-cell platform, and relatively few potential antigens of interest. And I think the reason is that the work that has been done to date has been somewhat disappointing and really, I would say, frustrating with respect to trying to achieve any sort of measurable responses in adults or children, particularly GD2 neuroblastoma, but adults or children with solid malignancies. In fact, it is in children with neuroblastoma targeting GD2 where anyone has really shown any sort of efficacy, but even that wasn't particularly high. And as you can see with several of the other selected targets, the response rates have been frustratingly low, I would say. And so why? Well, the challenges that are posed by solid tumors for adoptive cell therapy are as follows. The first one is trafficking and penetration. While adoptively transferred T cells are quite good at getting into the bone marrow and certainly the blood, they do find it increasingly difficult to home and extravasate and penetrate into the solid tumor microenvironment. Once they get there, the challenges that those cells face are active immunosuppression by malignant cells or their immune or the immune tumor microenvironment, induction of exhaustion, metabolic challenges, which result in poor proliferation and persistence. There is diminished or inappropriately low antigen presentation, and generally an environment that is not favorable to effective T cell function. And then even if they manage to supersede all those challenges, the adoptively transferred cells still are faced with the problem of target antigen heterogeneity, which of course is a source of inherent resistance, as Dr. Chen was explaining before in her presentation. And that can lead and does lead to antigen negative relapse if your T cells are good enough and function well enough. And so when we started this this project a while ago now. This was my first grad student, actually. And I was sitting in the lab and saying, let's take a step back and think about what type of immune cell might be better suited than T cells to the solid tumor microenvironment. And what we came up with was myeloid cells, and specifically macrophages. And the reason we thought that made some sense is because if you look at data like this, there are several data sets, but this is highly representative from hundreds of different patients with different types of solid malignancies. And you simply look at the breakdown of immune cells in the tumor microenvironment, what is clear is that it's not T cells, certainly not CD8 cells that predominate therein, but rather myeloid cells and specifically macrophages. And the reason for that is represented here and basically summed up as solid tumors like macrophages, because they can be easily subverted by the tumor microenvironment to become immunosuppressive or pro-tumor. Why? Because others have shown that myeloid cells like macrophages or myeloid-derived suppressor cells, but let's just call them all tumor-associated macrophages, TANs, help the solid tumors to metastasize, to invade or intravasate. They actually do participate in resistance to therapy, can maintain cancer stem cells, can provide a negative or immunosuppressive signal. Conversely, can cause cancer inflammation, and those, of course, aren't necessarily mutually exclusive, just depends on what sort of inflammation, and promote angiogenesis. And so all those things are highly desired if one were to anthropomorphize a cancer, highly desired by the cancer, and then and therefore help to recruit those myeloid cells through elaboration of various chemokines. And indeed, here's a question as to how. How is it that macrophages are drawn into, or monocyte and then macrophages are drawn into the solid tumor? Well, it's chemokines and chemokine receptors. We tested that experimentally. I will introduce CAR macrophages in a second, but basically we were able to look at cells that we made that ultimately we called CAR macrophages, as well as so-called M1 or so-called M2 macrophages, and using flow cytometry determined the pattern of expression of a variety of different chemokine receptors. And you can see that a variety of them are present there. And while data is not shown here, certainly more so than on T cells. And then we labeled some human macrophages with an IR-labeled dye, infrared-labeled dye, and injected them into immunodeficient mice that had previously been engrafted with a tumor. And then we dissected out the tumor, lung, spleen, and liver from here's a representative mouse. And the signal indicates an increased number of macrophages that have been that have homed into those sites. And you can see certainly some macrophages are to be found, some adoptively transferred macrophages are to be found in the spleen and the lung and the liver, but especially in the tumor. And so that gave us some confidence that at least the very first challenge of access and a productive immune response in the tumor macrophages might actually be quite suitable for that. There are other ways to deploy or use the potential functions of macrophages, as people in the audience probably know. Some of them are removing a suppressive signal called the SERP alpha CD47 axis. You can think of this as a macrophage checkpoint, at least how I think of it. You can try and block some of the chemokines or cytokines like CCL2 or CSF1 respectively. Those have their own challenges that we don't have time to talk about today. But the point of this particular slide is to show that there is a growing interest in macrophage-based therapeutics. And so what we sought to devise and to show is that engineered macrophages could exert multiple effective functions. So number one, trafficking, like I showed you. Number two, a specific killing function, which is phagocytosis for professional phagocytes, such as macrophages. And then immune activation, because after all, these cells are professional antigen presenting cells. And so we thought that we might be able to show that upon internalization of a pathogen, or more specifically here of a cancer cell, the macrophages will be able to break down that cell, present an antigen to T cells, and also activate an immune response. And that's what I'll show you momentarily. There were some challenges that we had to resolve first. the standard lenti or retro did not work. The VSVG-based lentivirus does not work very well at all for transducing myeloid cells, human myeloid cells. We tried a variety of ways, but eventually we came upon a chimeric adenoviral vector, not AAV, certainly not a lenti or retro, but an adenoviral vector called Ad5F35, which we actually found was very efficient at transducing human macrophages. So what we then ended up with is an ability to transduce human monocytes or macrophages to express at very high levels a transgene, a chimeric antigen receptor in this instance. And we have done a lot of other work showing that we can make those cells produce other molecules as well. So here are some videos that I will show you, and I'll play them side by side. Firstly, control macrophages on the left. Secondly, anti-HER2 redirected CAR macrophages on the right. And the target cells have been labeled with a pH-sensitive dye that glows green upon lowering of the pH, that is to say, phagocytosis into the phagolysosome. And so what you can see at the end of these videos, which, as you can see, last just over a day, is that control macrophages do not internalize cells, whereas the anti-HER2 redirected CAR macrophages do internalize these cancer cells. And that was so let me fast forward from there. Now, sort of more traditional assay can be done. I'm showing you here phagocytosis on the left and target cell growth. I guess you could think of that as the reciprocal or inverse of that on the right, where we used a HER2 expressing breast cancer cell line and exposed that either to CAR macrophages redirected to HER2. That's in the, if you look first on the left, that's the squares, the yellow squares. Or to macrophages, control macrophages, that is to say that they were activated in a similar fashion, but they did not have a CAR. And we redirected the macrophages against HER2 using Herceptin or trastuzumab, an anti-HER2 monoclonal antibody. And what you can see with the phagocytosis assay is that the CAR macrophages were much better at phagocytosing than macrophages redirected solely with an antibody alone. And that was really important for us to demonstrate. And the same thing, but in reciprocal, was shown on the right, where tumor cell growth is inhibited predominantly or most effectively by CAR macrophages, whereas if you use Herceptin, trastuzumab, or another anti-HER2 antibody, pertuzumab, those results weren't as good. So we then moved into an in vivo model using immunodeficient mice engrafted with a human cancer cell line that expresses HER2, treated either with phosphate buffered saline control or untransduced control macrophages or CAR macrophages, and followed those mice out over time. And you can see that all of the mice had a tumor, and so the control blue line mice died. The mice that received macrophages without any redirection had slightly more prolonged survival, but really it was the CAR macrophage recipient mice that had particularly prolonged survival, and some of them had no evidence of disease out to more than 100 days. And we were interested, we did several different in vivo models, but the work is published now, and so I won't go into all of those. But an important question that we asked ourselves, and that we have subsequently been asked as well, of course, is what would happen to those macrophages when they go into the tumor microenvironment? Would they have a dominant effect on the already existing resident tumor-associated macrophages there, or would they themselves be subverted back to an immunosuppressive phenotype? So and we did this in a number of different ways, but basically here is a highly reductionist experiment whereby we took control macrophages in blue or CAR macrophages in red. We took the supernatant from those cultures and put that supernatant with all of the soluble factors, chemokines, etc., and cytokines that exist therein, put that supernatant onto macrophages that we had previously polarized into a tumor-associated macrophage-like phenotype, an M2A or M2C phenotype. And we measured initially on the left, very simply, CD80 expression by flow cytometry, which is very simple, highly simplistic, of course, but nonetheless is correlated with a pro or anti-tumor phenotype, and showed that exposure to soluble factors elaborated by the CAR macrophages actually renders TAMs, if you like, or M2s into a anti-tumor phenotype. And in more detail, we then did RNA sequencing on those and found some of the findings that you see on the right side of the slide, whereby in the immunosuppressive macrophages, we actually find that they're influenced by CAR macrophages to become more inflammatory. Th1 pathway, INOS signaling, et cetera, were found in those macrophages. And then the question, of course, is what do our macrophages do to T cells? Well, we've looked in a number of different ways. Here are two on the slide. On the left is the chemotactic index of either resting T cells or activated T cells when they're exposed to conditioned media from control macrophages or CAR macrophages. And in both of those situations, exposure to CAR conditioned media leads to an enhancement of chemotaxis by T cells. And a more traditional immunology assay being shown on the right side of the slide is where we look for antigen-specific proliferation of T cells. So let me take you how this is done. We first made anti-NY-ESO-specific T cells and labeled those with CFSE, which upon dilution correlates with proliferation of T cells. And then we used either media in blue or control macrophages in red or CAR macrophages in green, as the control groups, and the experimental groups were either control macrophages that were loaded with the NY-ESO peptide. We knew that they had the appropriate HLA type, or CAR macrophages that were loaded with the NY-ESO peptide. And then what we sought to find out is whether the CAR macrophages would be better, worse, or the same as control macrophages when it comes to stimulating T cell proliferation. And I think the result is quite clear. And so all of the data that I showed you so far was with human cells and or in xenograft models. We then turned to doing some immunocompetent mouse experiments in order to study in greater detail the effect in an immunocompetent setting when there are T cells, dendritic cells, et cetera, on the effect of these macrophages. And this work was done not in my lab, but rather in the company in Charisma, and therefore the data is provided by Michael Kuczynski and Stefano Pirini. So these are BALB/c mice that were engrafted with a CT26 cell line that overexpresses HER2 and in the flank. And then we treat them either with nothing, untreated, or with UTD, which is untransduced macrophages in blue, or with CAR macrophages in red, measure tumor volume over time up to 60 days. You can see that all of the mice that are treated with control macrophages progress, except one actually, one resolved the tumor spontaneously, whereas the majority of mice treated with anti-HER2 CAR macrophages are controlled, all except 3 control the disease at long term. And then there is a survival advantage as shown in the bottom left of the slide. And you can see that the number of animals treated, 9, 9, and 8 in the different groups, and how many were alive at day 75. So that suggests to us that there is also in this immunocompetent mouse model the same effect that we showed previously in a xenograft model. And to delve a little bit further, we then took out the tumors from these mice and initially, again, very simply looked at the number and morphology of the infiltrating T cells. We stained this with an anti-murin CD3. You can see that there are many more T cells in the tumors that were in mice that were treated with CAR macrophages on the right compared to the mice that were treated with control macrophages on the left. And furthermore, and this is very sort of gross morphology, I suppose, you can see that those T cells are larger and more polarized and hence more activated. And then we did the key experiment, which is in the mice that had cleared their tumor after being treated with anti-HER2 CAR macrophages. We then challenged them with the same tumor cell line, except that was not overexpressing HER2. And here this is really the key experiment when it comes to epitope spreading and stimulation of a broader immune response. Perhaps you might characterize it as a vaccine-like effect as well as what we have shown before. So the BALB/c mice with the CT26 HER2 tumor that had been treated with CAR macrophages and attained a complete response were then re-challenged with a CT26 wild type, that is to say HER2 negative tumor. And controls were naive BALB/c mice that had not had a tumor or macrophages before. And then you can see the tumor volume over time at the top and then the survival at the bottom. And again, the results are really clear, which is that the CAR macrophage recipients challenged with an antigen negative tumor rejected that tumor. And to us, this supports a vaccine-like effect as well. The next question was, can we do even better? And so there's certainly a variety of, you can talk about a variety of different ways to try and improve that, but something that made a lot of sense to us, given the interplay with T cells, was to combine this approach with PD-1 blockade in the immunocompetent mouse model. So same setup, same tumor, but addition of an anti-PD-1, either by itself in black or CAR macrophages with anti-PD-1 in green. You can see that the survival is superior in the combination treated mice. And then dissecting out the tumors and looking at the absolute number of CD4 and CD8 cells, the absolute number of antigen-specific T cells by using a GP70 tetramer staining, or indeed of TNF production, shows a trend. In some cases it's significant, but largely I think a sort of stepwise trend as you go from no treatment to control macrophage to anti-PD-1 to CAR macrophage to combination CAR macrophage with anti-PD-1, an increase in the sort of cells that you want to see in the tumor. So to finish off, I will tell you that that whole process was developed as a GMP process, a good manufacturing practice process, so we could treat patients. The way this happens is we get our cancer patients, they undergo GCSF mobilization, not GM-CSF, but GCSF mobilization. Their peripheral blood mononuclear cells are apheresed in the usual fashion for CAR T cells, but then undergo a bead-based CD14 positive monocyte selection. Those monocytes are differentiated to macrophages using GM-CSF in vitro and become, by many different parameters, macrophages that are then transduced with the Ad5F35 vector that encodes the CAR transgene. And then what we are left with is CAR macrophages after a 7-day manufacturing process. And that can be, that product can be frozen and then thawed at will and injected into patients. And the yield, we have seen up to about 5 billion CAR macrophages. And this is partially automated using the Miltenyi Prodigy system. So earlier this year, we launched the company launched at Penn and UNC and several other centers are opening at the moment, a phase 1/2 clinical trial that uses autologous HER2-specific CAR macrophages in patients, adult patients with recurrent or metastatic solid tumors, not breast cancer necessarily. It can be any solid tumor that overexpresses CAR macrophage, that overexpresses HER2, with the intent to treat up to 18 patients. They undergo the process that I explained before in the previous slide. And we have two groups. The first one is currently recruiting with a split dose on day 1, 3, and 5. And the next group after that, next group of patients will be all of those, as long as found to be safe, will be all of the macrophages will be injected on the 1 day, but where the first bag of the macrophages will actually, the cells will be labeled with zirconium-89. That will allow us to study the trafficking and persistence of the cells in vivo in human beings with cancer, which will be really interesting when we get to that cohort. And very interestingly, I think the patients undergo tumor biopsy before infusion, tumor biopsy early after infusion, and tumor biopsy at around 4 weeks after infusion. We've already treated 2 patients, and the results will be presented at SITC next month. So to conclude, I think of engineered macrophages as a Trojan horse, because just like in this story, where the Trojans thought they were letting into their city a gift, or they were letting into their city something that was benign and even desirable to them. And it wasn't really until that night that the horse broke open and the Greek soldiers came out, is exactly the same concept that we think of for engineered macrophages, which will follow the same chemokine signals that tumors elaborate in order to draw into themselves what they think will be pro-tumor macrophages, but rather they are drawing into themselves anti-tumor CAR macrophages. And to acknowledge members of my lab, especially Mike Kliczynski, who was, as I mentioned, my first grad student, who then went on, co-founded the company with me and is the VP of research at the company, and then various funding sources that we relied upon for the first proof of concept of this technology. Thank you.
Thank you, Sa, for an exciting talk. There are several questions, and And we should probably start with, have you looked at, in your preclinical model, at persistence of macrophages after infusion?
Yeah, thank you for that question. We have. So I can tell you that in the the immunodeficient mice, we found that those macrophages persisted for around between 2 and 4 weeks. So these aren't T cells that don't proliferate at all, actually, and they certainly don't proliferate massively like T cells do, and they do not persist like T cells do. And when we first started this work, we weren't sure that we would see anything more than a effect for 2 to 4 weeks. But what made us quite excited was the potential vaccine-like effect, because that is how you can engender long-term responses, even if the cell that you infuse does not last forever.
Then there are, in the end, 3 questions around kind of the epitope or antigen spreading. So one is, have you, in the end, more carefully looked what actually the immune system is responding to, which is expressed by CT26? The second question is, have you looked how long these immune responses last? And then the third question is, have you kind of had a control group where, you know, you had tumors and then you just surgically resected them and then went back with tumor cells to see if there were either rejected or grew. So
very good.
I think they all touch on the same kind of, because I think everyone is in the end looking for it. You know, how can we achieve reliably antigen spreading?
Yeah. Great questions. Thank you. We haven't done all of that yet, and I completely agree. Those are the right experiments to do.
Then have you? I believe most of your studies have focused on, targeting HER2. Have you kind of explored other CARs in your macrophages targeting other antigens?
Yeah. The predominance of the work, certainly the work when it was in my academic lab largely was HER2 as a proof of concept, but we also developed anti-mesothelin and anti-PSMA CAR macrophages. We haven't done GD2. And so, but I mean, I see no reason why any CAR T cell target could not be just easily plonked into a macrophage.
Then there's a question in regards to brain tumors. Have you kind of explored that approach, for, brain tumors in preclinical models?
Yes, the company has. I haven't. And so, yes, there is a collaboration with an academic group to do brain tumors through regional injection of the macrophage, and I think that's a really great question, because again, it speaks to, if you were to ask me just a priori which types of tumors would you think should be treated with this compared to, say, T cells, I think, if my hypothesis is correct, I think that it is tumors that prove to you that they're good at recruiting macrophages. And so, for example, you might take a biopsy and see there's a lot of macrophages. That's the one to go for. And at least with GBM, I'm not overly familiar with brain tumors of different sorts, but at least with GBM, there's a lot of reason to think, because there's a lot of microglia there, at least, that might actually work quite well.
Then there's a question about kind of, side effects. Are you worried about kind of, CRS or ICANS, kind of side effects of kind of all, which are associated with other immunotherapies, but in particular for CAR macrophages?
Yeah, I think that, again, a priori, I do not expect that to be the case. You see CRS and subsequently neurotoxicity in CAR T recipients where there is massive proliferation of the T cells. And yes, we think that largely the IL-6 that is produced is actually produced by myeloid cells, not the T cells themselves in the patient. We're only putting the number of CAR macrophages we're putting into the patient is a tiny proportion of their whole myeloid compartment. And so I don't necessarily expect that to be the case. And I can tell you, semi-informally, that we've not seen cytokine release syndrome.
All right, I think before we go into the Q&A, since it's almost at the top of the hour, I just would like, please, Xar, and you won't stay on, but I would like to thank both of you for your wonderful talks. And I would like to officially close the event and also to invite all who are interested to stay on for continued discussions. Thank you all for coming, and please join us next week, October 14th, when Bradley Bernstein from the Dana-Farber will be presenting on epigenetics, mechanism of tumor genesis. I also would like to highlight that we'll have another Rising Star session on November 11th, which will feature Dr. Mariella Philbin of the Dana-Farber Cancer Institute and our own Gail Robinson's, who will present on pediatric brain tumor work. So to kind of, we should probably have a few more questions for you, and then there are still some other questions for you. One question is, what about, I know, let's call it conditioning regimen.
Yes.
Do you think there is a role of using chemotherapeutics to get rid of, let's say, immunosuppressive macrophages before you give yours?
Great question. Have thought a lot about this over the years. We haven't tested it yet, certainly not in patients, and the clinical trial that is currently ongoing does not use any sort of depletion. But yes, I actually think it would make a lot of sense to try and deplete tumor-resident macrophages. Question is with what? I don't think it should be fludarabine cyclophosphamide necessarily. A macrophage specific, to me, the most elegant would be a macrophage specific agent. Perhaps, I don't think these have regulatory approval, commercial approval, but a CSF1 inhibitor or anti-CSF1 monoclonal antibody, to me, makes sense, because I imagine what would happen, again, we haven't tested it, but I imagine what would happen is that there would be an increase in, for example, CCL2 production by the tumor, therein further improving the chemotaxis of the transferred macrophages if you timed it right. So that is to be tested.
There's one question in regards to manufacturing, and the question is, do you actually expand the macrophages, during this week, or do you in the end just select, differentiate them, transduce them, and then infuse them back? Could you comment?
We lose about 50%. So it's the opposite of T cells, where you might start with, X and you end up with many-fold X, depending on the time in manufacture. We actually end up with fewer cells since they don't proliferate, and there's various losses, etc. It's not a huge loss. It's, let's say, 50%. But they don't expand. No.
And then probably one other question is, as you know, in the T cell world, we are all worried that, previous therapies kind of have beaten up T cells. So what is kind of do you have any thoughts about, previous chemotherapy or radiation in the concept of macrophage fitness?
Yeah. So look, I think that, and this is a little bit theoretical, but I think this is one potential advantage of working with cells that are very short-lived, really, or relatively short, certainly much less than T cells, is that they haven't, these particular monocytes from the blood have not been exposed to chemotherapy. Their ancestors have, to be sure, they're stem cells. But less theoretically and more practically, this is something I was really quite worried about before we started testing this in monocytes from actual patients, is many of the CD14 circulating cells in solid cancer patients are kind of MDSCs, the kind of suppressive cells anyway. And so we needed to, and actually satisfied ourselves that those cells, after exposure to the viral vector, and this is actually a point that nobody's asked about, but I'll tell you, which is unlike lentivirus, which we think of as relatively inert, other than carrying out its function of transgenesis. The viral vector we use is not inert at all, and it's a very strong inflammatory stimulus. And so I think that, not that we designed it that way, but a sort of handy, I would say serendipitous effect of having to use this viral vector is that it is a very pro-inflammatory stimulus, and therefore the monocytes or macrophages that we get at the end of the manufacturing process are nothing like myeloid-derived suppressor cells, and we've shown that phenotypically as well as functionally. I
have one question of my own, and that is, having worked with AD5 and F35 in the past in the context of dendritic cells, I assume when you infuse them, actually the macrophages still present adenovirus-derived capsid peptides. So are you worried about pre-existing adenovirus-specific immunity, in your patients, or have you kind of looked at adenovirus-specific T-cell responses?
Yeah, excellent question. Thank you. Yes, we were worried about it. We have, yes, there will be some adenoviral peptides shown using RNA-seq for what it's worth. We've found that the more immunodominant, I guess presumably the protein products, the immunodominant protein products of the transcripts that we're detecting are actually not present in the macrophage, not presented in the macrophages, but some are, and there will be some increase in anti-adenoviral antibody titer after infusion, and that's why we're not planning to do more than one dose, albeit fractionated over 5 days. But in terms of toxicity, we haven't, with the very few patients treated so far, we haven't found a problem with that.
So there's now a question which was actually asked after Yvonne's talk, but I think we should actually all three give our opinion. And, it's the well-known toxicity case, the 2 deaths on the UPenn study with a dominant negative receptor in the context of CAR. And I know that there is not a lot of information available but, the question is really, what does it mean to the field? Is it a CAR target, or was it the DNR? And I would like that in the end, everyone could kind of, we can start with SARS, since you're at the institution, I don't know, and then Yvonne can kind of give her thoughts.
Well, thank you for that. I as you know, it's a pretty big institution and a big sort of group. So I was not involved in that trial, and I really can't comment on it.
So Yvonne, what are your thoughts?
So I know as much as anyone who has read the press release. So we don't have insight into the detailed data. I think what I can say is that the DNR had been tested by more than one group. So Kath Baller's group, for example, had done extensive studies on the DNR. Their trial went on for 2 years and actually never saw any toxicity that resembles what T-Immunity had reported. On the other hand, the PSMA CAR has previously been reported to have severe toxicity cases. And so, again, based on the scanned information that is currently available, it seems plausible that the toxicity is a result of the PSMA rather than the DNR. Now, of course, there's the question of whether the DNR would have exacerbated toxicities that could have resulted from the PSMA CAR. And I think that's hard to comment on without having more information. Certainly in our own studies, right, we have a CAR that can do what the DNR does. And on top of that, it would trigger T cell activation. And so toxicity is something that we have thought about a lot. And I saw that there was a question about this as well. And so I think there are a couple of things to note in the TGF-beta CAR platform. First, the CAR does not respond to the latent form of TGF-beta. It only responds to the active form. And it's dose dependent, meaning you actually need a fairly high concentration of TGF-beta to activate the CAR. And so those two things combined together really restricts where the CAR can be activated. The CAR also actually cross-reacts against mirroring TGF-beta because human and mirroring TGF-beta are very highly conserved in sequence. And we have not seen any toxicity in our mouse models, either immunocompetent or NSG xenograft models. That's not directly attributable to the tumor growth as opposed to the TGF-beta CAR. And so far, we haven't seen any evidence that would suggest that the TGF-beta CAR would trigger any sort of systemic toxicity. With that said, as I mentioned, we have been in detailed discussions with clinical colleagues in terms of clinical translation for the GBM platform. And so we consider whether it would be prudent to include something like a suicide gene. Right. Now, this is something, I learn every time I talk to clinicians, right? In GBM, the overwhelming priority is efficacy because unfortunately the patient will die for sure if we don't provide something that is at least somewhat efficacious. Also in GBM, they routinely apply high-dose steroids to control inflammation. And so in that specific indication, the strategy, at least based on our consultation so far, would be to not put in the suicide gene, but when necessary, apply steroids to ablate the T cells. But I think that's a really important question. Of course, safety is something that we always have to consider in these clinical applications.
Perfect. Yeah, I just wanted to add that, we have also, together with CAS, given, virus-specific T cells modified with dominant negative TGF-beta receptors to NPC patients and have not seen, any overt toxicity which we could relate to the dominant negative receptor. There was one other question in regards to your work, Yvonne, in the immune competent model. Have you looked at, murine T cell persistence, in your studies?
This is referring to the TGF-beta CAR platform, I assume? Yeah. So we actually just started doing orthotopic brain tumor models in syngeneic mouse models. And those trials are ongoing, so it's a bit early for me to comment on the results. Because we inject directly into the brain, we don't actually typically see any substantial number of cells in the periphery when we do RO bleeds. And of course, it being in the brain, it's kind of hard for us to take time point samples where the T cells are expected to be. So we right now don't have really quantitative answers in terms of persistence in our in vivo models. In our in vitro experiments, we've certainly cultured these cells out for weeks, and we add TGF-beta every 2 days, and the cells will keep growing. And so that suggests to us that the T cells don't actually become functionally exhausted, even under repeated TGF-beta exposure. But in vivo, we have to see.
Perfect. Sa, there's one question for you, and that is the role of CD47 in, the CAR macrophage system. Have you kind of explored that or thinking about combinatorial, approaches?
Yeah, we did. It is at best additive. It's sometimes neutral or additive, but it is neither synergistic nor is it inhibitory. I guess you would expect it to be inhibitory. What I mean by that is that combining CD47 or SIRP alpha blockade with the CAR macrophages in appropriate models like I showed in my presentation enhances phagocytosis. I think if it was a synergistic, I would be more excited about it. But to me, 1 + 1 = 2 isn't necessarily that exciting. Not to say that it shouldn't be ultimately tested clinically, but I do think, and I'm not necessarily on the PD-1 bandwagon for everything, but I just think that in this particular situation, it does actually make sense to if you're going to add anything, add something that's going to further give you T cell activity rather than further enhance phagocytosis, because the phagocytosis is quite efficient already.
Then, since we are a pediatric institution, of course, the question comes up, CAR macrophages for pediatric patients. Do you think we will be able to make enough cells? As you mentioned, you lose quite a few during production.
Yeah. Well, we've obviously never tried. I think that the ways to enhance, solely from a very, very practical manufacturing point of view, giving GCSF does increase monocytes as well as neutrophils and stem cells. So you guys obviously do that anyway and all the time. A longer pheresis and larger volume pheresis, and of course how long and whatever. So blood volumes. So it would, I assume, scale, but again, we've never tested. So maybe you need fewer cells for a smaller person, a child. So I don't see any reason really why not, but it should be tested, of course.
I think there were 1 or 2 questions coming back to the clinical study, and one is, I think, which struck some listeners, was really the low rate of CRS or neurotoxicity, even in these patients who had quite impressive tumor load. Do you have kind of any, I know you're not a clinician, but, from your preclinical modeling, do you think these bispecific CAR T cells are different than, let's say, a standard monospecific CAR in this regard?
Yeah, I mean, that's a fantastic question. That's the question all of us want the answer to. How do you get high efficacy with little or no toxicity? And I can offer my speculation, and I have to say it's speculation at this point, right? So the only strong predictor of severe CRS that I'm aware of is high tumor burden. And yet our patients have very high tumor burdens, and we saw nothing more than grade 1 and no neurotoxicity. And I suspect there are a couple of things that contribute to that. The first thing is, as I mentioned, we're really biomolecular engineers, so we spend a lot of time optimizing that CAR construct to make sure that, first of all, it's a true OR gate, as in even if it lost one of the two antigens, it will still recognize the tumor cell efficiently. And the CAR T cells are very, very potent, such that we can give such a small dose, 50 million CAR positive cells is what most of our patients received, and wipe out all of those tumors. And I think a key point is that the tumor eradication actually happens very, very quickly. So our patient 1, for example, you could see tumor nodules like bulging lymph nodes. And you can see those lymph nodes go away within the first week. By flow, we can see it. And I think I showed one set of flow data, right? By day 7, most of the tumor's gone. By day 14, it's nearly completely gone. On the other hand, T cell expansion almost always peaks on day 14. And so what that means is we actually have a separation of peak tumor and peak T cell temporally. And I think that separation likely also contributes to the lack of toxicity, because when you have massive amounts of T cells in the patient, you don't have massive amounts of tumor to stimulate those T cells that then drives this immune overdrive, right? So I suspect that also contributes to the lack of toxicity, which is sort of a benefit of strong and fast efficacy up front. We also manufacture the cells such that it would be enriched in memory phenotypes. And that may also contribute to that, although my personal suspicion is that the memory phenotype contributes more to the long-term persistence of the T cells. So all of our patients are still in B-cell aplasia, including the one that's more than 21 months out. And so that would be what I've been thinking in terms of why, but really it's a key question, and it's something that we will continue to think about.
Great. Any other? Let me just scroll up and down. Oh, Sar, there's one technical question for you. Which serotype adenovirus did you use or is used for the immune competent studies? Because, Ad5F35 is more, tropic for human cells.
Yeah, this was done at the company, but I'm pretty sure it was the same one. It's
okay,
Ad5F35. Yeah.
All right. So there was one, and I think that probably would be a question to you both. So actually, one of our faculty from the imaging department asked, we use a lot of, luciferase, what about in animal models using MRI or other imaging modalities to really get an idea when we then image patients, is that, for instance, pseudoprogression or not? So what are kind of your thoughts of doing kind of more other than luciferase imaging in these kind of preclinical models to then also really get a better understanding when we then image our patients after immunotherapy, what it all means?
You want to go first, Yvonne?
Sure. I mean, I think that's a great question, because we actually had to think about that a lot for the GBM model. Because first of all, imaging the brain is a little more challenging because you have to go through the skull. Although in our syngeneic mouse models, we can see the signal just fine. But curiously, in human GBM models, sometimes we see the loss of luciferase signal, even though the tumor is clearly there. Because when you sack the animal, you look at the brain and you can see the brain is clearly not normal, and yet the luciferase signal goes away. And we've after speaking with several colleagues that work on GBM, it's actually quite a common pattern where luciferase transgene expression is just not stable, perhaps because glioblastoma cells can differentiate. And so we actually have been actively looking into MRI as an alternative imaging system. It's just unfortunately not as high throughput. It's substantially more expensive. It takes a lot more time to image each mouse. And so it's something that is not quite as scalable as luciferase imaging. I think the question is actually touching on something slightly different, which is in terms of applicability to human patients, right? To be able to see whether the mouse model is able to capture key elements of what would eventually happen in human patients. And I think that's a really valid point as well, right? Perhaps MRI or other imaging modalities, I'm certainly not an expert on imaging techniques, right? Could give useful information that we have just been completely unaware of because we just always do luciferase imaging. So I think that certainly is an interesting area to explore.
I agree, and I would add to that this is, to whoever asked that question, I think that is an area that is very ripe for exploration. There have been several attempts. There are several ways. Preclinically, it's not that difficult. Of course, luciferase, you could do a bunch of things. You could do MRI, people have. You can do various PET. But, the point is, how translatable are these approaches to humans? In T cells, well, let me go with macrophages, or at least cells that are not actively dividing. And that's why we're really quite excited about use of zirconium-89 with the Explorer PET, which I'm told there's only a couple in the country, but luckily we have one, which is essentially allows you to capture whole body very quickly, just like a PET scan, but with increased sensitivity. And given the longer half-life of zirconium than indium, the indium-111 being the more traditional radio tracer, it will give us a better spatial resolution, as well as the ability to follow those cells out to about 7 days, if not a little bit longer. And so to me, that's an extremely exciting thing that we should be able to get to on the clinical trial. But for T cells, since they do divide, labeling them with any sort of dye that is diluted with each cell division is probably not going to answer any questions beyond the first few hours, or maybe first day if you're lucky. And it's really important, especially for solid tumors, but honestly, incredibly interesting and important, I'm sure, because we probably will be very surprised at what we see. I don't know what it'll be, but I mean, we have a lot of assumptions that come from mouse models, that come from various things, but we just don't know what happens in humans. And so devising a technique, and I think ideally it would be genetically encoded, just like the car, receptor or enzyme that allows you to use some sort of tracer that you could inject, and people have developed a few of these approaches. That is the way forward with transgenic T cell technologies in humans, I think.
Great. And before we close, Yvonne, you mentioned kind of on the side, we infuse a memory type CAR T cell enriched product, and that triggers the question: how do you enrich for memory CAR T cells in your production?
Yeah, you can, first of all, we enrich for memory subtypes to begin with before we even activate the T cells. And of course, you try to figure out culture conditions in terms of your cytokine cocktails to maximize the retention of that memory phenotype. And in our products, of course, it varies from patient to patient. But usually there's a substantial fraction of the final cryopreserved product that is still in the central memory phenotype. And I think that likely plays a role in the persistence of the product.
Perfect. So in closing, I think I would like that you both kind of just give your thoughts on this kind of, there is this notion in the field, CAR T cell therapy will never work. Also, of course, SARS a little bit, but CAR T cell therapy will never work for solid tumors. There are prominent scientists in the field who say it will. By the time, it's effective, if it gets effective, we will get side effects. So I just wondered if you could kind of in closing give your kind of thoughts where you see the field is kind of going over, the next 5 years. How can we make it, these therapies effective, in a safe way?
Ladies first.
Well, thank you. So, I mean, I think as an engineer, I am taught to be very practical, and I'm usually the first one to point out all the problems with with a proposed solution. So I can certainly see where this idea of, you know, CAR T cells will never work for solid tumors is coming from, because there are a number of really important challenges, right? With that said, I think if you ask someone even 15 years ago, is a CAR ever going to work for anything? Most people's answer would be, no, you're crazy. And so I think, you know, we've learned to never say never and just have to focus on what the problems are and what kind of solutions can we come up with. So what are the problems? I think in solid tumors, there are a few categories of issues, right? The first thing is access. So unlike blood cancers, where the T cell is in the same physical space as the tumor, the T cell has to somehow find the solid tumor. Right? So how will the T cell get to the tumor in the first place, especially if the tumor is immunologically cold and it's not sending out chemokine signals, et cetera, to attract the T cells? A lot of people are working on that with various strategies, oncolytic viruses, direct injections of stimulatory molecules, et cetera. We'll see which one works out best. And the second part of access is, once the T cell gets to the tumor, can it infiltrate the tumor? Right? And that's linked to the second challenge, which I would describe as immunosuppression, right? That tumor microenvironment oftentimes is so hostile that even if a T cell gets there, it doesn't do anything useful. And that's where strategies like our TGF-beta CAR and many other people's proposed solutions comes into play. How do you booster the function of the T cells once they get to that solid tumor microenvironment? And then that will then facilitate the infiltration, et cetera. And there are many different aspects to that, involving both soluble factors as well as, you know, the stromal environment, et cetera, that one can think about, right? And then I think there's a different category of issues, which is, what antigen are you going to target? Okay, and this is where both toxicity and efficacy converge, right? Oftentimes, the antigen one can find is not tumor exclusive. So then we worry about off-tumor targeting, you know, with things like mesothelin, HER2, CEA. They're attractive antigens, but they just have these off-tumor toxicities that you really have to worry about. And oftentimes, in order to make it safe, we end up compromising the efficacy. And one way around that is to build these more complicated logic-gated systems. And the question there becomes, how do you make these logic gates work effectively? Because once you get too complicated, the system can break in so many different ways, and then the efficacy takes a hit, right? There's also the flip side of the problem. We talked about how GBM is heterogeneous. So how are you going to find an antigen that would allow us to hit every tumor cell, right? And so antigen selection remains a very challenging issue. And I think all of these challenges just take more ingenuity. I am still optimistic that we will continue to get better, even though I certainly am aware that it's not an easy problem to solve.
Perfect. And Sa?
I couldn't have said it better than Yvonne. I would add that, or to give a slightly different ending to it. Cars, love them. You know, that's what I do for a living. But I think, for my money, that it probably has to be a TCR based, transgenic TCR based, and ideally going after a disease driving mutation. And so, you know, I wonder sometimes when people say that, are they including or excluding transgenic TCR modalities in the CAR bucket? They're not CARs, but they're engineered T cell modality. And the first 2 points that Yvonne made are as true for TCRs as they are for CARs. But the last point, to me, I think if you start with that makes more sense than restricting yourself to going after a cell surface antigen, of which there's basically only one that I know of that is cancer specific.
Perfect. So I would like to thank you both again for your talks and for the good panel discussion. And I hope, and there's still 85 people on the WebEx. So at maximum,
how many of them are listening?
260. So I think you didn't bore anyone, and everyone was excited to listen to you and also listen to the discussion. So thank you again, and I hope that we see each other in person in the not too near future. Bye, everyone.
Thank you.
Thank
Thanks
you.
for the invitation. Bye.
Bye.